so much for coming to today's program. My name is Erin Payton, and I'm the Executive Director of the 19th Century Charitable Association, which is located in Oak Park, Illinois. Our mission is strengthening our community through learning, giving, and the sharing of our landmark building. We're so glad to have you at today's program, and it's going to be a really awesome, informative one. Before I bring on the first person, I just want to let you know that you are probably going to have some questions. And if you do, you can put them in our Q&A or in our chat. And if we have time at the end of the program, we will actually give you an opportunity to ask your question to the presenter yourself by unmuting you. So be ready with that and listen for your name. But right now, I am going to bring to the program Susan Roberts, who is on our science committee for our programs.
Hello, everybody. As Erin said, I'm on the science program committee, and we have a very exciting guest today who is our speaker. Besides being gorgeous, she is extremely bright and an expert on nanotechnology, which is something I really would like to learn about. So I hope you'll join me in this enthusiasm to learn about bioengineering and nanotechnology. Now, Tolou Shokuhfar is Associate Professor at the UIC and a director of their In-Situ Nanomedicine Lab in UIC Bioengineering Department. In talking with her, she's really a woman of the world. She got her undergraduate degree at Sharif University of Technology in Tehran. Then she got her master's degree in Portugal. She met her husband in a conference in Alaska and got her PhD at Michigan Technology Institute. So she has really seen a lot of the world and had a lot of wonderful experiences. So I'd like to introduce Tolou Shokuhfar, and I look forward to your program.
Hello, everybody. Thank you, Susan and Erin, for your kind introduction. It's a great pleasure to be here, and it's a great pleasure to be able to talk a little bit about some of my work and some of my research and give this talk to everybody. And I appreciate your time this afternoon. So if you would allow me, I'd like to share some slides with you. Well, today I'm going to talk about nanotechnology and what it is and why is it important. So let's start with this. Actually, one day I was in the mall and I thought to myself, let's see like how many people really have heard about nanotechnology. It was interesting because many people ran away. They found like, oh, what she's talking about. Is she crazy? Some people knew that, well, it is something really tiny, something that no one can see with their naked eye. Two of them had absolutely no idea. And there was a person who knew that 1 nanosecond is 10 to the -9 second. So that's a general thought of like what people in general know. Now let's give another example. Let's have a closer idea of what nano could mean. Let's think about what has a football to do with nanotechnology. Well, if we want to compare the soccer ball Earth and a viral capsid, we can think about a soccer ball to an Earth, the size of a soccer ball to an Earth, and is similar to the size of a virus to the soccer ball. So meaning that if we compare it to each other, a soccer ball is 10 to the power 9 bigger than the size of a virus. So size of a virus is in the scale of nano. So if you want to imagine how small is a virus, just imagine how small is a soccer ball in compare to the size of the Earth. That's how small we are talking about. Another example is your skin. Now, take a look at your hand. And now, take a look at the smallest wrinkles you can see in your hand. So the size of your hand is actually 10 centimeters. Now, if you take a look at the smallest wrinkles you can see with your eyes in your hand, skin of your hand, that's about 1 centimeter. Now you look closer, we're talking about 1 millimeter. This is by far what you can see with your naked eye. Now, if you have to go look closer, you go down to 100 microns. This is the size you can see with a light microscope. Now, if you're going smaller scale and you zoom in into that wrinkles, you can see the size of a white blood cell, 10 microns. Then you zoom in into that size of the white blood cell, you zoom in that white blood cell, and then you can see 1 micron. And then in that size, you're not able anymore to see anything with the light microscope because it's not possible. Then if you even zoom in closer, now you have to use electron microscopes, and then you can see DNAs. And then here we are talking about nanoscale. So the size of the DNAs are in the nanometers. So we are talking about 10 nanometers. So basically, if we want to imagine if a nanometer were the size of a penny, a foot would stretch from Miami to Seattle. So this is an example of the degrees of smallness. We have nano in nature. So let's imagine some of the examples that we can find in nature. So things in nature, now scaling examples, if we think about them. So an ant is about 5 millimeter. Then we have dust mites. Now they're smaller than millimeter. We're talking about 200 micrometers. So nanometer is actually 1 million millimeter. Now think about a fly ash. Fly ash is about 10 to 20 micrometer. So we are in the world of micro. So micrometer is actually 1,000 nanometer. So red blood cells are in the world of micro. Now, if you go down to scale, we have proteins and ATP synthesis. Now we are going down to nanoscale. And our DNA is between 2 to half a nanometer in diameter. Now we are in the scale of nano. And single individual atoms, for example, the ones that we see here on the bottom of the image, are the atoms of silicon. And their spacing is 0.78 nanometer. Now we are down to the scale of angstrom. So we can see actual individual atoms. You see them here. These are taken by electron microscope. And then you can see individual atoms, but they are below nanometer. So this is how small they get. What are nanomaterials? So what we talked about previously in previous image, basically, when we go back to nanoscale from proteins to DNA and atoms, now we are talking about materials, matters that are in the scale of nano, 10 to the minus 9. But the definition is actually very interesting because now when we go to that size, everything changes. The properties changes. Materials that are in that scale, materials that are in the scale of 10 to the minus 9 meter have different properties than bulk materials. Nanomaterials are those which are defined as having unique physical properties that are driven from inherent nanoscale dimensions of the material. These properties are the result of large surface area to volume ratios inherent at the nanoscale, which drives novel quantum mechanical traits. So a primary example is the change in the electronic properties of the material, which we call it quantum size effect, which is defined as unusual properties of extremely small crystals that arise from confinement of electrons to small regions of space in 1, 2, or 3 dimensions. Meaning that when we go down to nanoscale, we can have materials called nanomaterials that are either one-dimensional, two-dimensional, or three-dimensional. The result of quantum effects often yields new mechanical and even catalytical properties. So once we are at this nanoscale, their properties changes. This is an example of a nanomaterial we can find in nature. I believe many of you have seen lotus, the lotus leaf. But have you ever thought about it, that why lotus leaf never gets wet? It's very interesting. Lotus actually has a self-cleaning effect. Lotus leaf never gets dirty. But why? Once we put the lotus leaf under the electron microscope, we can see the surface of it has some bumps. These bumps will not allow the water droplets to get absorbed and sit on the surface. And actually, the water droplets will roll. And by rolling onto these bumps, they actually remove the particles of dirt. So the droplets here do not slide, they roll, and particles are removed if the force of absorption are higher than static frictions between the particle and the surface. So that's why the lotus leaf has this self-cleaning property. It's very interesting to know that historically, nanotechnology has been used for many years ago. Like what you can see here, Lycurgus cup in many centuries, like 800 BC, had the technology of nano. It changed its color by the liquid that was poured into it. It was green, changed it from green to red, depending on what liquid it had. And the reason was there is an interaction of light with gold, silver, copper, and iron nanoparticles. It became green because of the fact that silver nanoparticles are around 40 nanometer, and therefore they reflected light. And they become red because gold nanoparticles of 20 to 40 nanometer transmitted the light, and it became purple because because copper nanoparticle will also transmitted the light. So how did it work? It's interesting. Let's watch this video in YouTube.
An ancient color-changing artifact. Learn about it next on today's Creation Moment. And now, here's our Creation Moments host, Ian Taylor.
One of the most interesting artifacts at the British Museum is the Lycurgus cup, a beautiful goblet made by the Romans 1600 years ago. The cup depicts the story of King Lycurgus, entangled in grapevines for his treachery against
I'm not sure
Dionysus,
if you can
the
hear
god
this
of wine in Greek
voice,
mythology.
but
it's not the design that makes the cup of such great interest to scientists. When viewing the cup in normal lighting, the glass is jade green. But when the cup is lit from behind, the glass changes to bright ruby red. What is this ancient sorcery that gives the cup its color-changing properties? It's the precise amount of silver and gold nanoparticles dispersed throughout the glass material. New research has discovered that the cup even changes colors when liquid is poured into it. Scientists replicated the makeup of the cup and then filled it with various liquids. Their results suggest the cup might have displayed many different colors depending on what sort of beverage was poured into it. As one science website put it, quote, "Researchers are just now, all these years later, learning about such color-changing properties of materials with embedded nanoparticles. The hope is that these properties can be exploited to perform chemistry or medical tests cheaply and quickly by displaying different colors under different conditions. However far you go back into history, the ingenuity of man points to the even greater ingenuity of his creator."
so we can see that even at ancient Rome, they used nanotechnology and nanomaterials, nanoparticles, but they probably really didn't know the science and technology behind it. So in 2000, the government had a few definitions for nanotechnology. National Science and Technology Council Committee on Technology and Subcommittee on Nanoscience, Engineering and Technology defined it as research and technology development aimed to understand and control matter at dimensions of approximately 1 to 100 nanometer. That was defined as nanoscale, or the ability to understand, create, and use structures, devices, and systems that have fundamentally new properties and functions because of their nanoscale structure. But now let's think about a new terminology, nanomedicine. What is nanomedicine? Nanomedicine is actually the application of nanoscience and nanotechnology in the field of medicine to facilitate its diagnosis and treatment of human diseases. So we can see that nanoscience and nanotechnology and nanomaterials have recently found their way in the field of medicine. Let's hear about some interesting stories. Can it enable imminent immortality? So a funny and interesting story was that in 2005, Kurzweil writes of millions of blood cell-sized robots, which he calls nanobots, that will keep us forever young by swarming through the body, repairing bones, muscle, arteries, and brain cells. Improvements to our genetic coding will be downloaded via internet. And he predicted that, it was 2005, and in 2005, he predicted that in 20 years, that might be a reality, basically meaning that in 5 years from today. But in my personal opinion, we're getting more closer to death what we can see from this pandemic, but it doesn't mean that this would never become a reality. Immortality, I wouldn't say necessarily, but the fact that we can probably combat many diseases, that might be a reality. Well, there were stories actually, and many Hollywood movies coming out, such as in Spider-Man, the nanotechnology scientist, or also the Hulk, which was created by nanotechnology. These all seems like sci-fi or sci-fi movies. If you recall or have heard about the Fantastic Voyage in 1966, also a General Electric TV commercial in 2005, they talked about injectable nanobots. So he actually was kind of maybe inspired by these. But is this something just as a dream or a sci-fi movie? Or can we think about this as something that could inspire the field of nanomedicine, or something that maybe one day can become close to reality? And where do we stand today? How far are we from this today? Actually, I would want to say that not too far. According to Professor Richard Smollett from Rice University, he was a Nobel laureate in chemistry in 1996. He actually said, well, how soon will we see the nanometer scale robots that was envisioned by Eric Drexler and other molecular nanotechnologies become a reality? He thought it would never become a reality. But where we are today, this is where we are today. And not actually today, basically, in 2015. This was 5 years ago. We are much more progress today. It was published in 2015 that injectable nanomachines could reach any lymph node metastasis. And this is only one example. We have many more publications out there. So you could see like a lymphatic system and a systemic targeting of lymph node metastasis through the blood vascular system by using size-controlled nanocarriers. So you can inject systemically these nanomachines that could reach any lymph node metastasis regardless of anatomical position. So what are these nanomachines? So these nanomachines are actually nanoparticles. They are nanoparticles that are precisely controlled in size. They contain an anti-cancer drug core, which is protected by a shell. It is called a polymeric micelle. The whole particle, the core and the shell together, are 70 nanometer in size. And the core itself is 30 nanometer in size. So it's a size modulated polymeric micelle for targeting lymph node metastases. Once it's injected, you can see in the far right side images, these are fluorescent microscopy images. From top to bottom, the top image is green. It is projecting and showing the 30 nanometer particles. So it means that in that area with T, which is the tumor, the 30 nanometer particles have reached that tumor area. The red one is the micelle. So the core, which is the drug, together with the shell, so they have not been separated, both had reached the tumor area. The blue is a node, lymph node. So it's definitely in that lymph node. And the final image, which is the merged image, showing that the particles have reached the lymph node. So it's a merged colored image showing that the particles are in the right area, right part of the body that they should be. So we are not too far away from really being able to reach that idea, that dream. These nanomachines may not look like actual machines or robots. They look like these particles, but they are doing the job. In other simple terms, we do have nanotechnology-based products already in the market, such as wound dressings that are powerful antimicrobial barrier protections. They can maintain safe bactericidal concentrations of silver with patented nanocrystalline technology. Or we can have toothpastes with silver nanoparticles in them that can enable prevention of germs and bad breath. In 2007, was also nanofabrics produced that can deactivate bacteria and viruses and also break down harmful components of air pollution. Well, it's interesting because today we really need these clothes. But what is the focus of my research? So my research is in the field of nanomedicine. And I focus on total joint replacement and bone implants. I'm sure that many of you either have or know about people who do have bone implants. So bone implants are the implants that come in direct contact with hard tissue or bone in our body. They include dental implants and total joint replacements, such as hip implant, knee implant, shoulder implants. So what is the aim of these implants? And what are the problems related to these implants? So these implants are usually made out of metals because they are load bearing. They have to bear the main load of the tissue. And they are because they are replacing a load bearing tissue, they're replacing the musculoskeleton, they are replacing the bone. But what happens is that there are many loads applied. And these loads are applied at the interface of that implant with the bone. Once these loads are applied, that interface may become weakened. And what happens when that interface weakens? Infection can happen and the implant can fail. So there is 90% failure of the implant due to the loosening of that implant. And the loosening is mainly because there is not enough bone implant integration. We call this bone implant integration osseointegration. So from here now, when I mention osseointegration, what I mean is that interdigitation or interlock between the bone and the implant. We want that interlock and interaction between the bone and the implant to be as strong as possible. We want that interface to be very strong without any loosening. because once that loosening happens, then the bacteria and germs and all the bad things will happen, and then the cause of the loosening, and then what will happen is that you would need to have a revision surgery. So there are many reasons that this loosening can happen and then cause dislocation, wear of articular bare surfaces, and mechanical dislocation. So now let's look at the history of bone implants. And since we mentioned that surface, that interface between the bone and the implant is what is determinant for the survival of the implant, everything, all the important properties go back to the property of the surface of the implant. So the surface of the implant is the most important part of the implant, is the determinant part of the survival of the implant. So the evolution of the bone implant surface technology is what has been changed over the centuries. So from 1960s, when the first generation of implants were introduced, they were introduced as bioinert implants, where they elicit minimal toxic response, and the choice of materials match only the mechanical properties of the bone. So they only designed the materials and the implants to bear the load. Therefore, the materials of choice were metals and alloys. Then in 1980s to 2000s, the second generations were introduced, where the surfaces become more bioactive. So bioactive and or biodegradable. So these type of surfaces elicit a controlled physiological response and or degrade over time for eventual replacement by host tissue. In this stage, ceramics, polymers, and surface modifications and coatings were introduced. And most recently, the third generation, which were biomimetic surfaces, were introduced. So what do we mean by biomimetic surfaces? These are the surfaces that mimic properties of the bone on the nanoscale and or attempt to guide a control response using nano or molecular cues. So the best surface is the surface that can mimic the properties of the bone as closely as possible. So these are the next generation surfaces. And why is that important? Well, if we want to replace a bone, we would want to replace it with something that is as close as possible to the actual structure of the bone. So let's look at the structure of the bone itself. So the bone is that macrostructure, contains cortical bone and cancellous bone, as you can see here. So we have cortical bone and cancellous bone. In the area of the cortical bone, we have osteons. The osteons are at between 10 to 500 micrometers. So we have the microstructure of the bone. It's at 10 to 500 micrometer scale. Then osteons itself contain Haversian canal, and the Haversian canal contain lamella. Now we are talking about 3 to 7 micrometer size scale. Then the lamella itself contains collagen fibers. We are now down to nanostructure. Each collagen fiber contains collagen fibril. Each collagen fibril is 0.5 micrometer. And collagen fibril just themselves contain collagen molecules and bone crystals. And now we are talking about 1 nanometer scale. So when an implant comes in contact with the bone, so right after the surgery, when an implant comes in contact with the bone, what happens is the first things that it sees is the red blood cells, the whole body fluid, protein, and cells. And they have to come and sit on the surface of the implant and start forming and making all these bone crystals and collagen molecules, and then make the whole, make these bone crystals and collagen molecules and cause the reverse of these and basically make the actual bone. So which surface will enable this process to happen faster? Of course, a surface that is in this size scale, a surface that can trigger the formation of bone crystal and collagen molecules faster, and enable for a surface to enable this faster, it has to be at the same size scale. That's why when you look at a orthopedic surface, you see that this is roughened. This is not smooth, because when it's roughened, then it enables the cells to adhere to it better and then to function better and start promoting and releasing molecules that can produce bone crystals and collagen molecule and then the whole bone tissue. But older type of surfaces and surface modifications were just coatings that were at the micro scale and or just doing sandblasting. But now we are making much more organized surface changes. We call them nano or nanotubes because they do look like nanotubes. So these are these tubular structures that they do look like the shape of the collagen molecule, collagen fibrils, and bone crystals. So what happens when we have this implant and the surface looks like that nanostructure, the cells here can come and adhere on these surfaces faster because first of all, the genes see these nanotubes easier and the proteins can adhere on these surfaces faster, and therefore the cells for their adhesion on the surface require the protein and focal adhesions, and therefore they stick. And the cells, when they stick, they can have a better functionality, they can proliferate and differentiate and then become tissue. And it is not only limited to one type of implant. We can have these surfaces on shoulder implants, on ankle implant, on knee implant, on hip implant, on spine implants, on any type of bone implant. Well, interestingly, it is good to know that, well, what is the process and how fast we can get this down to the market? For any type of biomedical device, as you all know, we need FDA approval. And an FDA approval for biomedical devices is a long process. But according to FDA rules, if a device is already used in the medical field and you just want to make a change, then you don't need to get approval for all the process. You just need the approval for the new modification to that process. And we are doing a small change to make a huge impact. So We want this to get out to the market fast. And the process is not that hugely challenging or difficult. We talk about nano, well, we are not going to require any heavy equipment or expensive equipment and challenging manufacturing stages. What we are doing is simple. It's a simple electrochemical anodization process. It's possible to do it in a kitchen counter, but we are not going to do it in a kitchen counter because we have to use hydrofluoric acid, which can burn your bone down. Well, we have to do it sometimes in the clean room, but the process itself is simple. So you have a cathode and you have an anode, and then you have a beaker, and you have that secret electrolyte that contains fluoride ions. Fluoride ions are the ions that actually can etch metal. So titanium itself has a native oxide on the surface, and due to the electrochemical field and the presence of fluoride, oxide gets thicker and fluoride ions can diffuse through that oxide. So due to the diffusion and oxidation that happens simultaneously, these nanotubes form on the surface. So basically, these nanotubes are not an added coating, but an alteration of the native oxide. So they are they are biocompatible and they are scalable. So you can see scanning electron microscopy images of these nanotubes. In A, they are top images of the nanotubes. B, the bottom images of the nanotubes. C and D are side images of the nanotubes. So you can see that in A and in B, each single nanotube is 100 nanometer. These are very small scale nanotubes. When a surgeon sees a device that has been modified with nanotubes, they wouldn't see a change in the device. They wouldn't even notice there is a difference in the orthopedic implant. They will just use their same surgical process. However, these nanotubes can make a big impact. So this is a process by which the nanotubes are formed. I'm not going to go through that detail, but it's interestingly, we can form them in the shape of the actual bone. So in the right-hand side here, you can see the iliac crest bone of a 19-year-old donor, and here you can see the nanotubes. And they are very similar to an actual bone. So the impact they have is when we look at a single cell on the nanotube surface and we zoom in, we can see the paw or the feet or the hand of the nanotube of the cell that uses to adhere on the substrate and grow is actually going inside, attaching to each individual nanotube and growing inside the nanotubes. Here, it basically looks like it's growing inside the nanotube. And why this is important? Because once the cells are better adhered, they can better proliferate and differentiate and function better. So they have a better adhesion. The tissue will better adhere. They have better osseointegration. And for us to really see how that interface between the cell and the substrate is, we use a focused ion beam of gallium and cut the cell and the substrate and looked at that interface. And we could see that actually they are growing inside the nanotubes. So since I mentioned osseointegration is an important factor, integration of the bone with the implant is an important factor for the success of the implant. we wanted to test the success of the nanotubes with integrating the implant with the bone. In order to do that, we do in vitro, meaning in the laboratory tests of osseointegration. So the samples that were tested were polished, meaning that only a surface of the implant without any treatment, acid treated, meaning just a little bit, dipping it in the acid, just cleaning it. Surfaces that exhibited nanotubes that the diameter of the nanotubes were 80 nanometer, and the surfaces that the diameter of the nanotubes were 30 nanometer. And we could see that the cases that exhibit that had nanotubes on their surfaces exhibit genes and proteins that were indication of osseointegration, versus when there were no nanotubes on the surface, those genes were not exhibited. The same in these images were shown. So the genes osseopontin and osteocalcin, also DAPI, and actin expression, all of these are indication that the nanotubes were enhancing the attachment of the bone to the implant. And again, we counted the number of cells on different surfaces that exhibited, that had nanotubes on them, and the number of cells on the surfaces that had nanotubes compared to the surfaces that did not have nanotubes were much higher. In an animal study also, in a 4-month animal study, in a 4-week animal study, the new bone formation was much higher when there was nanotube around the implant compared to when there was no nanotube around the implant. So meaning that if you have an implant with these nanotubes on the surface, the healing would be much faster. So you can have your bone integrating and also integrating with the implant within 4 weeks, versus when you have a regular implant, this may take months. And these were also further results showing the same thing. This is a mouse that the implants were implanted. So one of them had nanotube, the other did not have nanotube on the surface. And again, we can see much higher bone formation in the case of the nanotubes. And after the animal was sacrificed, we compared and could see that the histology that was conducted ex vivo showed a considerably greater new bone formation with the nanotubes in the same animal. And this was done in 8 animals, and they all showed the same results. So you can see here, this is the new bone that is formed. And this is the implant which has nanotubes. Here there is no nanotubes, and we don't even see any new bone formation. So this was a 12-week study. So in 12-week study, if you have an implant that there is no nanotube on the surface, and then you have another implant that has nanotube on its surface, within 12 weeks, the implant with nanotube can make your bone grow faster and heal faster, versus the implant which does not have nanotube will not have any bone growth yet. So you have to wait a couple of more months for it to heal. Now, another question may arise is that, well, how strong is it? Is it gonna come off? Maybe if it's a dental implant, is it I'm gonna chew a nut and it just like loosen? Well, or is it are these nanotubes gonna come off during insertion of this during surgery? So there are certain tests that could be done, and that's a pull-out test. And so they pulled out these implants which had nanotubes on them, and they could see that the nanotubes sticked, and average pull-out force were about 3 times higher than controls, meaning that when it had nanotube on the surface, it required much more power and force to remove the implant from the tissue compared to when there was no nanotubes on the surface. This tells us 2 things. One is that the nanotubes make the implant stick to the bone much stronger, and the other one is that they don't come off from the surface. So these were as showing all the good properties for osseointegration that the nanotubes can provide. So we know now that the nanotubes can enhance the integration of the bone and bone growth on the surface of the implant. But what about multifunctionality? Excitingly, again, as I mentioned, not only the nanotubes can enhance and cause the bone to heal faster, but we can use them as carriers of drugs. So we can load the nanotube with different drugs. Here we load them with naproxen sodium and anti-inflammatory drug, and then we can have it released over time. And therefore, if you have like growth factor, antibacterial drug, antimicrobial drug, anti-inflammatory drug, you don't actually then need to take them orally. These will have a sustained release of very small amount of the drug, and then it can prevent an infection, which is one of the big causes of implant failure. So how infection happens and why we have this bacteria resistance and why we have a lot of these problems related to antibiotics. So the reason that infection happens is like you can have either floating bacteria that can come and sit on the surface, and when it sits on the surface, it expands and forms a slime, and then slime becomes biofilm. And then they become resistant because they communicate with each other and then change their gene and then tell each other to become resistant. And you can have also floating bacteria and host cells and proteins. This is like in the case of surgery, in the site of the surgery. But if you have a surface that is not a good host for the floating bacteria, then the cells come and sit and there is no place for bacteria to adhere. Or if you have a surface that has no reaction with bacteria, then like this has these nanotubes on the surface that do not react with bacteria, the bacteria will not have a chance to come and sit on the surface. So you basically are preventing this window of opportunity. And that's what we're doing here. So the nanotubes will actually not allow the bacteria to come and sit on the surface. So once we tested the polished surfaces, meaning the surfaces without nanotube, compared to the surfaces with nanotube, we could see that the surfaces without nanotube had much higher number of bacteria compared to certain surfaces with nanotube. So these green ones are the surfaces with nanotube, these green and yellow ones. These are the surfaces that have nanotubes. And this black one and red ones are the ones that don't have nanotubes. And the graph shows the number of bacteria. So basically here, the green dots are bacteria. And you can see in this case, 80 nanometer nanotubes, 80 nanometer nanotubes present the minimum amount of bacteria. So nanotubes do have antimicrobial properties as well. And we have enhanced that by adding silver oxide nanoparticles to the nanotube. So we can actually have silver oxide nanoparticles within the walls of nanotube to prevent any bacteria adhesion. And we got the same result as increasing the silver concentration can increase the number of dead bacteria and decrease the number of live bacteria by 99.9%. And why that would be important? Because it's not only limited to bone implants, but unfortunately, any type of implant that contains metal and metal alloys, such as pacemakers, can be infected. But in the case of pacemakers, this infection can be deadly. As you can see here, this pacemaker is made of titanium and is infected, and then it can cause really, really bad outcomes. So we took this pacemaker and we formed a nanotube on its surface. And you can see with naked eye, when you look at it, it's just a small change in the color. But when we put it inside the electron microscope, you can see these small nanotubes that are forming on the surface. However, it's just a small change in the color in the case of pacemaker. And then the cells are pretty healthy and happy when we look at them and culture them with the cells. And they do not actually come off from the surface. They are sticking on the surface, so they're not peeling off. When we do the insertion and removal test, they do not peel off. Even if they do, like if we intentionally remove them to see what happens if we do remove them. So we remove them and make a powder out of them and feed them to the cells like this. So we culture them with cells. If the cells even eat them, nothing will happen to them. They still function very well. So we found them inside the cells, but the cells were still alive. And the reason is that once they're introduced into the cells, into the body fluid, a corona forms around them. That corona is made out of proteins. So it's like a Trojan horse-like thing, that this protein corona is a biophysical corona that kind of mimics the structures and proteins that cell is familiar with. So it basically, this corona is made of calcium and phosphate and other proteins, so it doesn't make it toxic. So finally, what we have is a next generation nano-engineered orthopedic implant, meaning that with a small change on the surface of this implant, we have made a big impact by improving that interface. So we have enhanced its osseointegration. We made sure that it's not coming off and it's survivable, and it prevents stress shielding. I didn't talk about that, but it doesn't prevent stress shielding. It's antimicrobial, and it can have a prolonged drug release. We have a patent on this, or some news articles out of it. And these are my funding acknowledgements and my group. So thank you so very much for your attention, and I'll be happy to answer any questions you have.
I feel like just in general 10% smarter after watching that presentation. So thank you.
Thank you very much.
It wasn't too hard to get me a little bit smarter. Well, we do have some questions, and the first one I am going to attempt to unmute is Agatha Gallo.
Thank you so much, Dr. Sok. I don't know how to say your last name. I'm sorry.
Shokuhfar.
Thank you. It was a fascinating presentation. I really, really learned a lot, like others, I'm sure. But my question is, most of this has been done on, tell me if I'm right, on animals and donated bone, right? And I was just wondering when, you know, how was the donor bone accessed? And when do you think these devices will be available after FDA approval? I mean, it sounds like it's very important to be able to move this ahead.
Thank you so much. So these, till whatever I presented was my work, and they were done in the laboratory at the university. So we do have like a donor bank available by the university, and there were like funding agencies that enable funding that research. But then I had licensed it to a company. That company is moving it forward, and it's like doing right now a clinical trial study on dental implants. So once that clinical trials take a long time, like a couple of years. So I believe once that clinical trial is completed, then this can come out. So I'm hearing about it, and it's kind of in a good stage. So it wouldn't be too long, I would say. Maybe I would say less than 5 years.
That's great. Thank you.
Thank you.
Joyce Porter, you had a question?
Yes. If they're sending something to attack a tumor, how do the nanoparticles find that particular tumor and not attack anything else? Thank you.
Well, that's a very good question. So with the nanoparticles, if you recall, we talked about that shell around them. So that is not just a simple shell. Within that shell, we have functionalized groups, and we have like different peptides and antibodies and many other things that could be on that surface, designed on that surface, that only will go and stick to that specific tumor and cannot stick to anything else. There are other type of nanoparticles, for example, magnetic nanoparticles that could be injected into the spine and use specific magnets to be controlled and then be directed toward the part of a like tumor that they have to go. But they are usually designed in a way that they can only move toward that particular area that the tumor is. Wow. And the nanotoxicity aspect of them is also been investigated. So they wouldn't, like, if part of them gets to a certain organ, their size and concentration is controlled in a way that they have tested and shown that they wouldn't do anything to that specific organ, because they're designed only to release the drug once they get to that organ of their interest. Because for, say, tumor say a brain tumor the tumor of the brain has different property to the actual healthy tissue of the brain. Its pH is different, its elasticity is different, all the properties is different, and these particles are designed to start to release that drug not in a healthy pH, but in a tumor pH, like in a more acidic pH or in a more elastic property tissue. Say these are different in them.
Thank you.
Thank you.
Marie Cruz, I got some more questions. You have definitely opened a pipeline. Marie Cruz has a question.
I am wondering, when you do get a hip replacement, how do you know you're getting nanotech surface?
You would never know. Even the doctor wouldn't know.
Wow.
You're kidding. That's not good.
You have to choose, like the doctor can say like, well, I prefer this type of implant because of my patient, and then they will discuss it with the patient and say, would you like to have this type of implant or not, the old ones? So I think that's how it would probably go. You wouldn't know.
So you don't know exactly what you're getting, every single ingredient in the hip.
No, because even you look at it, they don't look different. They are so small. They don't feel different. They don't look different. They just function different.
That makes sense. Jonathan Kelly, if you can unmute yourself, you can ask a question.
I would say they probably would know, because down there at the beginning you wouldn't know, but you would basically know if you heal faster.
There you go. If you're climbing Mount Kilimanjaro, you got the nanotech. All right, Jonathan Kelly is, oh, here he is. Hi. Okay, I'm going to ask for him. Is this nanomedicine technology now in use successfully in other countries?
Other countries are working on this. Nanomedicine is worldwide field, and many scientists and communities are working on this on different areas. Yes.
Okay. Any in particular kind of leading the pack, or other than us?
So yes, Germany, France, China, they are doing the
great.
Yeah.
Nan Brulette, did you want to ask your second question that you put in?
Yes, this is fascinating, very far beyond me, but I'm wondering, what is your background? How you got into this field? What did you study?
That's a good question, Nan. Thank you.
Sure, thank you. Well, how did I got interested in this? My dad is a professor and my mom is a medical doctor. She's a gynecologist. So I needed to make them both happy. So I decided to become.
Is that possible? If it is, teach me how to do that.
So I how to become an engineer and
okay.
Medical field. I decided to become a bioengineer, but that's how I become interested in both. By training, I'm a material science engineer, so that's how I got my bachelor degree. My PhD degree is in mechanical engineering, biomechanics. That's how I got interested in orthopedic implants. And then my postdoc is in nanotechnology, postdoctoral degree. And that's how I got interested in nanomedicine. And now I'm a faculty in bioengineering. So that's how everything
So it's just kind of a ladder approach. You got one place and got another interest and got another. That's really interesting. Yes. Okay, because of the time, we're going to ask one more question. And that question is going to come from Gilherm. If you want to unmute yourself.
This was great. Thank you very much. I was curious about buckyballs too, but we don't have time for that. I'm curious, you kind of walked us through 3 generations of evolution or development. What's your guess as to what the 4th generation will look like?
I think the main aspect would be, well, I didn't say that, maybe the name probably should be next generation instead of 3rd generation, because the ultimate goal would be to mimic the natural tissue. So the closer we get to the natural tissue, the closer we will have the success of any type of material or device that we want to replace the tissue with. So I would say the next generation will be those biomimetic devices or implants that closely or as closely as possible can mimic the tissue that they want to replace it with. So if we are here right now that we are mimicking the scale, maybe the generation after this would be mimicking the function as well, and mimicking like the 3D bioprinting. That's what it is trying to do. So we're trying to mimic the growth. Maybe we try to mimic the growth of vascularization. Maybe we make a bone itself, not making just the bone to grow, making it the bone from the bulk itself, from top to bottom, from bottom to top, like from molecular stages.
Well, thank you so much. This again has been an amazing presentation, and everyone's brain is a little bit bigger now. Do you have any last things you want to add before we close?
I just want to say how grateful I am to be here, and I'm very thankful for your interest. I hope that I could talk to you in a way that brought some interest. And if there is anything else I can do at any time, and any questions or interest if anybody had, I'll be happy to help or talk about. So thank you so much for your attention.
Thank you, Tolou. It was so nice to meet you. And I just want to thank everyone for coming out today. We have these programs every single Monday, except for during short break in the holiday season at 1:30. If you want to know more about our upcoming programs, visit our website, www.19thcentury.org. If you want to help us keep putting on these amazing programs, if you'd like to make a donation, we would love to accept it. This is the time of giving, and I believe tomorrow might be Giving Tuesday. And so it'd be a great time to visit our website, www.19thcentury.org, and click the Donate Here button and drop us a few dollars. Well, for everyone at the 19th Century Charitable Association, this is Erin Payton. And again, thank you to Tolou and to Susan and everyone else for making such a great program. Have a great day and happy holidays. Bye-bye.